Hello, my name is Aidan Bergeron. I will be presenting my project on the synapse and its relevancy, its functions, and its importance. The topics that I will be covering in this project will be what is the synapse, the presynaptic events, the postsynaptic events, what happens at the synapse, the diffusion, degradation, and reuptake of the neurons, synaptic integration, and the many varieties of synapses that take place. To begin with, what is a synapse? The synapse is the site where the neurons communicate with other cells, where action potentials arrive at axon terminals of the neurons that causes the chemical messengers known as neurotransmitters to release. After they're released, the neurotransmitters cross a tiny gap that is approximately 20 to 40 nanometers wide. After they cross a tiny gap called the synaptic cleft. The neurotransmitters that bind to the receptors on other neurons or other types of cells, such as muscles, glands, and some organs. The diagram to the right is a image uploaded by Simply Psychology showing the procedure, the diagram, and what is in the synapse, some of which we will be looking at more so into this project. This is a visual image of the presynaptic events by the Encyclopedia Britannica, showing the series of events that go within the presynaptic events from start to end. We will be looking more in depth into what the presynaptic events are in the coming up slides. Within the synapse, and as previously mentioned, in the presynaptic events, there are two kinds of vesicles. The synaptic vesicles, which contain small molecule neurotransmitters. They are also produced in the soma of neurons, which is the center. Neurosecretory granules, which contain the large molecule neurotransmitters. These types of neurotransporters are then transported down towards the axon terminals, moving the materials with them from soma to axon. These materials are proteins, which are called transporters, and their job is to carry the molecules across the cellular membranes, loading up the synaptic vesicles with the necessary small molecule neurotransmitters, which are then synthesized, meaning to combine so to form a new complex product. These neurotransmitters that are synthesized by molecules that were already there and with the help of synthetic enzymes. After this process with the synaptic vesicles, they move towards the active zones, where they release their stored neurotransmitters in a process that is called docking. Calcium ions that are extra, more so than what was needed to enter the axon terminals, causes the synaptic vesicles to fuse to the membranes and release the contents of those neurotransmitters towards the synaptic cleft through a process called exocytosis, where they are taken out exported out of a cell. These neurotransmitters that are then exported outside of a cell are recovered through a reverse process called endocytosis, which the molecules are taken into a cell and given a new neurotransmitter. The process that the larger molecule neurotransmitters are released is similar. However, the difference between them is that the large molecule neurotransmitters are synthesized and processed by endoplasmic reticulum, which is the membrane system that has multiple functions, and the synthesis, modification, and transport of proteins. Now, looking at the postsynaptic events, the receptors, mainly the ionotropic receptors, are located across the neuronal membrane, when they're bound by neurotransmitters. Ion channels then open or close. The ion channels that react to the binding of neurotransmitters are known as neurotransmitter-gated ion channels. When these ion-gated channels become binded channels, metabotropic receptors are embedded directly into the postsynaptic membrane. Instead of forming on channels, these indirectly affect a cell's function in which it triggers various events inside the cell. When a neurotransmitter is attached to the metabotropic receptor, a protein within the cell is activated. This protein is called G protein, which its full scientific name is the guanine nucleotide binding protein. The receptors which grab these G proteins are called G protein coupled receptors. The G proteins then use a shortcut pathway where the activated G proteins then bind to ion channels inside the cells, causing it to open. This channel is then referenced as a G protein gated. On the presynaptic axon terminal, there are receptors in which their job is to regulate the release of neurotransmitters by presynaptic neurons through a negative feedback loop. Autoreceptors send out an alarm when the neurotransmitter levels are too high in the synaptic cleft. The extra neurotransmitters are bound to the autoreceptors, which then tells presynaptic neurons to stop releasing neurotransmitters. What happens at the synapse afterwards? When neurotransmitters bind to transmitter-gated ionotropic receptors, the receptors are then tasked with either opening or closing by changing its shape to best complete the task it needs to do. The ionotropic receptors control various ion channels that contain chemicals such as Na+, which are sodium ions, and Cl-, which are chloride ions. When binding a neurotransmitter to a Na+ channel, it transports those sodium ions to the neuron where it causes it to depolarize. When neurotransmitter binds to receptors on the Cl channel, the neurons instead hyperpolarize. Neurotransmitters that are released into the synaptic cleft can only stay there for a short amount of time, which has been registered as about a few milliseconds to a few seconds. The reason this happens is due to the receptors no longer working. They become unresponsive due to the overstimulation, which could cause communication errors among the neurons and cells. Preventing such cases and the prolonged activation of these receptors are through mechanisms within the synapse called diffusion, degradation, and reuptake. When neurotransmitters are removed from the synaptic cleft by the diffusion, it diffuses those neurotransmitters towards the extracellular fluid where it's absorbed by the glial cells. After the neurotransmitters are diffused from the receptors, the neurotransmitters are then degraded by enzymes called monoamine oxidase, or MAO, turning the transmitters into a class known as monoamines. Another neurotransmitter called acetylcholine is degraded in the synaptic cleft, turning it into choline, an acetic acid, by the acetylcholinesterase enzyme. Neurotransmitters can also be reuptaken by the same neuron that released them. This occurs with the help of transporters, which are proteins that forms across channels across the cell membrane. Next, we have the synaptic integration, where neurotransmitters that bind themselves to a receptor results in the opening of Na+ channels, which causes it to depolarize the neural membrane, which is called the excitatory neurotransmitter. And a neurotransmitter that binds itself to the receptors that opens the Cl- channels causes it to start the hyperpolarization of the neuronal membrane, which is called the inhibitory neurotransmitter. Within a single neuron, it can receive thousands of inputs from other neurons. Some of the inputs they receive are dealt with and mediated by the excitatory neurotransmitters, which results in it forming excitatory synapses. The same neuron receiving excitatory neurons is also dealt by and mediated by inhibitory neurotransmitters, which causes inhibitory synapses. These two synapses, the excitatory and inhibitory, hyperpolarize the neuronal membrane, preventing it from firing any action potentials. In this case, it adds up both the synapses it gets from other neurons, and if there is more excitation within the neuron than there are inhibitory, the neuron will be more likely to fire an action potential. The process in which the neuron performs and determines if it will fire an action potential is called synaptic integration. When the neuronal membrane has a change in voltage due to the Na+ or Cl- entering into a neuron is called the postsynaptic potential, and it causes the voltage of the neuronal membrane to move towards the activation threshold, which is called EPSP, or the excitatory postsynaptic potential. And counter to that one, a postsynaptic potential that causes the voltage of neuronal membrane below the activation threshold is called a IPSP, which is inhibitory postsynaptic potential. And the diagram below shows an example of this. A postsynaptic potential can change the membrane voltage by around 1 millivolt. Therefore, EPSPs can add up to be capable of enough voltage to depolarize the neuronal membrane beyond the requirements of the activation threshold and can cause an action potential to happen, which is known as summation. And within summation, there are two types. There is temporal summation, where it occurs when several postsynaptic potentials follow each other with little delay and are piled together, added up at their arrival, and they arrive together at the synapse. This version of summation, spatial summation, occurs when several postsynaptic potentials occur at the same time, but unlike its temporal counterpart, they arrive at the same time. Spatial summation, they are added up together at different synapses. The varieties of a synapse, such as a synapse with a reflex arc, a stimulus such as putting your hand in a boiling pot causes the heat-sensitive Na+ channels on our sensory receptors to open, leading to a series of action potentials to go through the afferent pathway to our spine. Inside of the spinal cord, the action potentials that are created by the axons of the sensory neurons forming synapses with interneurons, which act as a middleman carrying the sensory information, turns them into synapses with motor neurons. The action potentials that started in the motor neurons travel through the efferent pathway towards the periphery, which causes actions like our muscles to contract, which in turn, with the previous example, lets us take our hand out of the boiling pot. And within the reflex arc contains two kinds of synapses. The polysynaptic, which is where the neuronal pathway, such as the reflex arc, includes more than one synapse, and they would use the polysynaptic pathway. And lastly, the monosynaptic is when a neuronal pathway only requires one synapse and nothing more. Between the other motor neuron and muscle cells is located the neuromuscular junction. One muscle cell is stimulated by only one motor neuron. The membranes on the muscle cells have deep slots known as junctional folds. The active zone of the axon terminals of the motor neurons line up above the slots, and in each slot contains multiple receptors and Na+ channels. Through an experiment performed by Lowy, where he experimented on a frog heart, he electrically stimulated the vagus nerve entering the frog's heart, which had been bathed in a fluid causing the heartbeat to slow down. He then proceeded to pump the fluid into a second frog's heart, causing the second frog's heart to also slow down. And through this experiment, discovered that the transmission of information between neurons were chemical and electrical, where because of this debate, they discovered electrical synapses afterwards through this experiment. These occur at sites called gap junctions, where the synapses are lined up together with proteins called connexons, which grants passage to the ions to travel towards one neuron to the other. To conclude my project, there are many functions and mechanisms working inside of our brains every day, and a better complete understanding on our brains could lead to possible interesting discoveries and ways to manipulate them for good intentions, for example, like antidepressants or any kind of brain issue. And by choosing of the synapses, I believe is relevant to our course, because across neuroscience, the synapse is relevant for transporting information between the cells. It is complex, it has many mechanisms, operations, and variables that it has to account for to make sure that nothing goes wrong within our brain and our body. Thank you for listening. Have a good rest of your day. This is the link in case any issues arise, that the link for the paper and the project did not go hand in hand.